Okay, good morning, grade 10s. Good morning, everyone. You all good? Okay, so yesterday we did the external structure of the leaf, and we spoke about all the various parts of the leaf. Today we're going to be looking at the internal structure. So this is basically the parts of the leaf that are actually performing the functions. And so this is really, really important for us to get into. We're going to see where those tissues are, what those tissues are doing, how they're getting their requirements, etc., etc. So yesterday, just quickly, with regard to the external structure of the leaf, a leaf is attached to the stem by the petiole. It's at that point that we have what we call an axillary bud. An axillary bud is where a new leaf will start to grow. So we have a petiole. That petiole is continuous with the midrib. The midrib is the main vein, and it will contain xylem and phloem, vascular tissue, to transport water as well as food to the leaf or away from the leaf. Obviously, food away from the leaf because food gets produced in leaves and needs to be stored in the roots and the stem. Okay, then we have the apex of the leaf and the base of the leaf, and this margin over here, which basically can either be smooth or it can be jagged or it can be lobed, doesn't really matter. But you can see those are all the structures of a leaf. The whole leaf blade is what we call the lamina. Okay, so that's what we did yesterday. Today, this is just a picture just showing you all the different types of margins that we can have and variation that exists within leaves, etc., etc. Okay? In most leaves, the dorsal, the upper part, so this is quite an important concept, grade 10s, is understanding when we talk about dorsal and ventral. Okay? So dorsal is the top surface of a leaf, ventral is the lower surface. If, for example, if I had a dog that was standing in front of me over here, the dog's back, that is the dorsal part of the dog, where his tummy is, that's ventral. Everyone get that? So dorsal, ventral. The way I always remember it is I know that D comes before V in the alphabet. So dorsal top, ventral bottom. Okay? Dorsal, ventral. Then, if I've still got my dog, just for reference sake in terms of biological terms that we use and stuff, anterior is from the front, so this is where my dog's head is. That's anterior, where his tail is posterior. So anterior, posterior, dorsal, ventral. Does everybody get that? Yes? Okay. So it's quite an important concept because when we talk about certain surfaces, you need to have an understanding of what we're actually talking about. Okay, so we're going to be talking about the dorsal side and the ventral side of the leaf. The dorsal side, the upper part of the leaf, is usually bright and shiny, while the ventral side is usually dull. You can distinguish between the dorsal and the ventral side of a leaf. If you pick a leaf, you can see which is the top and which is the bottom, right? Yes? Okay? The veins are also usually on the ventral side. So the veins are usually more prominent on the ventral side, and the dorsal side is usually quite flat. Okay? The reason why it is quite flat is so that it can obviously have greater exposure to light. Then we have, there will be more stomata that occur on the ventral side. Can anybody think why we have more stomata on the ventral surface as opposed to on the dorsal surface? Yes, come on, hello.
Mainly the dorsal surface that has more sunlight to heat it, and then the ventral side will deal with the gas.
Okay, so that's 100% correct. You just need to give me a little bit more. Okay, so the dorsal side gets direct exposure to sunlight. Correct. While the ventral side is dealing with gases. But why is a plant happier or why is a plant better suited to have more stomata on that side because of that exposure to light? What is it trying to do? What can be lost through our stomata pores that the leaf is trying to prevent loss of? Water loss, right? Everyone agree with me? What is the term we use for water loss again? Transpiration, okay? And so, the fact that we have, let's say this was the top surface of our leaf and this is the bottom surface, it's quite difficult to draw in terms of kind of understanding. Sunlight is hitting here, so this is light coming in, right? This is getting great exposure to sunlight. While our stomata, made up of our guard cells, are on the ventral surface, the lower surface, okay, they kind of are a bit shaded. Wow, the brain was slow there. They're a bit shaded, okay, which then prevents or decreases transpiration. Okay, so transpiration is the loss of water by plants. And so the fact that our stomata are on the ventral surface as opposed to on the dorsal surface actually is because of decreasing transpiration. Okay, pretty smart, because the top surface, that dorsal surface, actually shades the lower surface. And as Kamohelo said, these are responsible for gaseous exchange. The guard cells with the stomatal pool. Okay, that's all the external structures of the leaf. Is there any questions? Sorry, one more slide before that's all. In monocotyledonous plants, remember yesterday we spoke about dicotyledonous and monocotyledonous, yes? We know what that means? Yes? Cool. So in monocotyledonous plants, there are two differences. One we discussed yesterday. Their veins are parallel. They run parallel in the leaf, okay, in monocotyledonous plants. So we have a leaf, right? And the veins run parallel. That's the first difference that we learned yesterday. In dicotyledonous leaves, the veins is branched. They don't run parallel, they are branched out like that. Are we all happy? So in dicotyledonous, in dicotyledonous plants, they also have a petiole, where in monocotyledonous plants, the leaf basically attaches directly to the stem. It doesn't have a petiole. Are we happy with that? Think about a millie. You seen a millie? It just grows, those leaves just grow straight on the stem. Okay? They don't have this petiole. So in monocotyledonous plants, the petioles are absent, and the leaf blade is directly attached to the stem. These are known as sessile leaves. Okay, so they are attached directly to the stem. They don't have a petiole. So two differences between monocotyledonous and dicotyledonous leaves. Ashley, are you okay? Falling a little bit asleep? Or just drifting a little bit? Anyway, two differences. One, their veins. Can you see the difference in their veins? And two, the presence of a petiole in dicotyledonous plants as opposed to monocotyledonous plants that attach directly. Got an itch in my foot. You ever have that? Anyway, okay. Woo! You know what's the most uncomfortable thing on the whole planet? Is when your secret sock is so secret that it starts to become even secret to your shoe. Have you felt that? It like slips. Have you ever? You know? When it starts to slip, and then it gathers in that part of your... It is the most uncomfortable thing. I think it's just horrid. So, random little sentence there. Anyway, for those of you watching at home, I just want to give you the real life experience of what it's like to be in my random class. Those random things and soft conversations. We have random conversations, right? Yes. But it's fun. Well, I think so. I don't know, maybe you guys just think it's lame. Woo! That sanitizer has a very strong smell when you're close to it. Okay, let's get on to internal structure today. So there's all our external structure of a leaf. Let's go internal. Move in. Okay. So, if we examine a leaf, and now we're going to be specifically looking at a dicotyledon. Try again. We're going to specifically be looking at a dicotyledonous leaf in cross section. Okay. This is sometimes quite difficult. So what we're taking is a leaf that we are looking at, that you can see the dorsal surface and the ventral surface. And we're looking at the tissues in between. So a thin leaf, everybody get that? Okay? If we could take that thin leaf that we have in front of us, we then cut it, and then we're looking at it with the tissues inside. Yes? Who wants to quickly, Jessica, quickly run down there and go get me a leaf. Just a nice, preferably a nice big leaf, if you can find one. Those olive leaves are too small. Like, preferably, I don't know. See what you can find. So, because I just want to make sure that you understand this from a vision perspective. Yes? Okay? Right, so if we examine a dicotyledonous leaf in cross section from the dorsal side to the ventral side, so so dorsal to ventral, there are three main groups of tissues that can be distinguished. We have the upper and lower epidermis. So we've got the outer covering of the leaf called the epidermal layer. Okay? We already know about the epidermis, we know about the structure of the epidermis, but we're going to look at the epidermis in a bit of detail. Then we have what we call the mesophyll tissues. Now the mesophyll tissue is made up of the tissues that we've studied, the plant tissue. Specifically in a leaf, it's chlorenchyma. Okay, because chlorenchyma's got lots of chloroplasts. Okay? And we're going to speak about that mesophyll tissue. There are two main mesophyll tissues. That kind of makes the bulk of the tissue inside of this. Can you see it there? Yes? And then we have vascular tissue or conducting tissue. So we have the conducting tissue found inside of the vein within the mesophyll tissue. Okay, so there's three main sections that we're going to be looking at as we look at our leaf. I just want to make sure that Jessica isn't like lost. I want to carry on without her, but also I don't want to not keep going, you know. Okay, let's see when she comes back. Are we all good? Ah, here she comes. Good. Leaf hunting. Well done, Jessica. Very good leaf hunting. Look at those beautiful leaves. Look at those beautiful leaves. Monocotyledonous or dicotyledonous, guys? Is it in? Can we see? Can we see? Look at our leaves that Jessica went hunting for. Yeah. Okay, it is a. How do you know it's dicotyledonous? How do you know it's a dicotyledonous leaf? Because the veins are branched. Do you see? So we have a main vein and then we've got branching happening. Yes? You can see the dorsal surface to the ventral surface. Do you all agree? So dorsal's kind of shiny. It's got a cuticle. It feels a little bit waxy. Okay, we can't touch, but whatever. You can go find yourself a leaf, feel it. It's got a waxy cuticle. The cuticle does what? What does the cuticle do? Your turn to talk. The cuticle does what? It prevents water loss. What is the word for water loss, guys? It prevents transpiration. Okay? I'm coming back. Can you stay to my right here? Okay, I just want to. Okay, so we're going to take our leaf, right? And we are basically cutting our leaf in cross section so that we are looking at it like that. That's what we're looking at. I don't know, is it in? So we're looking at all these tissues. She's like, a bit hectic, I can't see anything. This is microscopic, guys. Everyone get that? So we're looking at the tissues inside of this beautiful thin leaf. And this is what we're going to see. So we're going to see an upper epidermis. Do you see an upper epidermis here? I mean, do you see the upper part? Yes. Okay, we're going to have a ventral epidermis. Yes. Okay, so ventral and we have dorsal. I wonder if this leaf is going to be friendly for me. I don't think it's going to be friendly for me. See if we can look at some stuff under a microscope at some point. Depending on if I can get a good specimen. This is not a good specimen to get a. Ooh, maybe. Maybe. Oh. Maybe we're going to do something. Maybe we'll look at something under a microscope. How does that sound? Yes, ma'am. Does that sound fun? Now the kids at home don't get this experience. Like, what's going on? What's happening? What's she doing? Hey? No, they must see. They must know. They must see. Choose the door. I'll prepare this anyway. Okay. So, what I'm going to do, I'm going to just try and remove a part of the lower epidermis and see if we can see some stomata. We might not. We might. Let's see what we can see. I mean, we've got the leaf here. Okay, so I'm going to switch off the video.